Dear ladies and gentlemen, it is my great pleasure to give you a short talk today on opportunities and limitations of research in the living collections of botanical gardens, based on experience here at Bonn University Botanical Gardens and on the special occasion of the 400th anniversary of Oxford Botanical Gardens. I will be going over a few of the topics and techniques that we have been using to work with our living collection, and I will start off with a very rough classification of living collections in botanical gardens. This is a typical collection which nearly all of us hold, which is a collection which looks like cactaceae, but as a matter of fact, they could also be reclassified. Botanical gardens are institutions holding documented living collections in order to fulfill their mission in scientific research and teaching, education, and in species and nature conservation, as Rauer wrote in 2000. And cactaceae tend to belong to a particular class of botanical garden collections, which are the so-called stand-aroundiaceae. There are also plants which we specifically hold for teaching or teachiaceae, for purposes of species conservation or conservaceae, and for display purposes such as the borders of roses or tulips, which I would call displayaceae. However, a fifth group of plants that we hold in botanical gardens and which may overlap with any of the other four is what is in the focus of my presentation today, and those are the so-called researchiaceae, the plants that we specifically do research on. One of the modern techniques, of course, the typical modern technique of working with plants is looking at their phylogeny with molecular techniques. And of course, living collections, if they are large, are a prime source of high quality DNA or RNA, however that be, for major phylogenetic studies. The quality of these phylogenetic studies depends on, of course, the quality of the sampling, which depends on collection size the study is based on, and they depend on DNA quality. Of course, the best DNA or RNA you can get is from actively growing living plants, and together with our Chinese colleagues and some colleagues from Kew, we were able to do a major lamate phylogeny a few years ago, which was largely based on the collections of Bonn University Botanical Gardens. We've got a very large spread across all orders and families of the angiosperms, so we were able to provide over 200 or more than 70% of the samples from Bonn University Botanical Gardens. This is, of course, no news to you. This is a very common use for botanical gardens collections, although the use of collections is, of course, today slightly compromised by the implementation of the Nagoya Protocol. One of the large research collections we are holding at Bonn University Botanical Gardens is the collection of Boraginales, or as some colleagues would like to call them, family Boraginaceae, here with a handful of typical representatives, typical Boraginaceae, but also Cordiaceae and Eritiaceae. And we've been doing a hell of a lot of different studies on them, phylogenetic ones, of course, but also a lot of morphological studies. Now, many of us are able to use an SEM, a scanning electron microscope, for their studies. And the SEM indeed permits an incredibly detailed look at plant surfaces and to a certain degree also at plant structures. So we did a major study on fruit evolution in Hydrophyllaceae, concentrating on the genus Phacelia. And you can see the ovary primordia on the upper left developing into fully functional ovaries with receptive stigmatic surfaces. And then on the right, you can see the mature fruit as they have opened and released their seed. So these pictures are all very nice, but what you do not see is the details of placentation that are happening on the inside. And with the advent of microcomputed X-ray tomography or micro-CT, we can now look into the structures we are studying non-destructively without the tedious process of sectioning, and can actually study both placentation and ovule seed arrangement in unprecedented detail non-destructively. And this, in this particular case, this led to major insights into multiple transitions from 4 to many seeded capsules and vice versa in the genus Phacelia, elucidating the phylogenetic pathways, trajectories leading from the one to the other, and helping to explain the whole phenomenon of seed number variability in angiosperms, or that is at least what we would like to believe. The fruits and ovaries can be studied with microcomputed CT, but another thing that is very beautiful is that you can look at what we call internal floral spaces. Now, if you look at the 2 flower pictures in the middle right, you can see 2 flowers of hydrophyllum, and you can see that they don't look exactly particularly exciting, but we can see that they've got these strange 5 bulges on their petals. And if you look on the insert on the left-hand side, you can see that the petals have strange flanges with papillination on the top, which run from the bottom of the corolla to the middle of the petal lobes, of the corolla lobes. Now, if you investigate the entire flower under micro-CT, instead of looking at the brutally open flower under the SEM, you can see that these strange flanges enclose nectar chambers, which are highlighted in blue on the lower right. And these nectar chambers differ dramatically in their volume and in their interconnectivity, one to the other. So they form a lobed nectar container which can be accessed by pollinators only at the very top, which you can see on the right-hand side. You can measure the volume, you can measure the dimensions, you can measure the interconnectivity, so you can suddenly have a whole set of insights which are of crucial importance to the functional floral morphology of these plants, and this is only possible with techniques such as micro-CT, where you study the flowers on the inside without cutting them up. A flower that you all know is Heliotropium corymbosum. You may know it under the name Heliotropium arborescens, a commonly cultivated, highly scented vanilla flower essentially from Peru. And the flowers from the outside, they don't actually look too exciting. You may wonder why anybody would want to study them, but if you put them under the micro-CT, you can see that they are incredibly complex in their internal structure. So from looking at a plant or a destroyed flower from the outside, you suddenly get an understanding of what happens on the inside of that flower. What you can beautifully see here is the corolla tube with the anthers inserted in the middle of the tube, essentially closing the tube apart from a minute pore in the middle. And underneath that opening, you can see the complex style stigma complex, which is characteristic of the family Heliotropiaceae, as we would call them, with an stigmatic ring and a sterile apex, and the whole structure based on a long style, a thin style, which of course is not receptive nor secretory. So you can look on the inside of flowers and start to understand the evolution in plant groups such as the Boraginales, but you can also study the complex flowers of plants such as Loasaceae, which are, for various reasons, very rarely kept in botanical gardens, but very difficult to study in the wild because they are usually very narrowly endemic and very rare in nature. If you're lucky enough to have them growing, you can study the internal morphology of the flowers, which is extremely intricate, with 5 floral scales made up from staminodia, which you can see on the right-hand side. And if you analyze the digital images you get from the micro-CT, you can identify the area where the nectar is presented, the so-called nectar container at the base of these floral scales, and you can see the guidance structures here highlighted in green, which direct the hummingbird's beak during pollination, during flower visits, to the nectar container at the base of the flower. So there's a whole lot of things that you can suddenly investigate by using novel technologies. Many things you want to study in flowers are of course dependent on the fact that they are still living. And this is very difficult in the field with plant groups such as Loasaceae, which are widespread but very localized and very rare and highly seasonal. So it would be very convenient to have them all growing in your greenhouse and do your studies in situ instead of having to go to South America and hoping that it has rained. This is Nasa macrophersa, one of the most easily cultivated species, and Loasaceae have got, this group Loasaceae has a very peculiar phenomenon, and that is thigmonastic pollen presentation or stamen movement, which means that if a pollinator visits the flower, the fact of the pollinator visit, the physical manipulation, leads to pollen presentation. There are approximately 100 stamens, and they are sequentially presented upon a flower visitor. And we had a huge research collection of Loasaceae growing in our greenhouses, approximately 60 species of the relevant subgroup. And you can see two of my former PhD students here playing with the flowers, mimicking pollinator visits. And we got such a large data set that we can now show that not only do many of the plants react to pollinator visits, but we can even show a clear evolutionary trajectory with an increasing complexity and speed of reaction upon pollinator visits within the phylogenetic progression of Loasaceae. This is the graph from that publication, so we can actually show, I think for the first time, not only that plants show behavior, but that this behavior shows an evolutionary trajectory. This is, of course, a very surprising result, and this is only possible if you actually have your plants growing in a scientific collection, because doing these experiments in the field, especially across roughly 50 to 60 species, would be not only too time consuming, but it would also be impossible to standardize experimental procedure in a way to actually trace evolutionary trajectories. Coming back to the borogenales, another thing that you can do with scanning electron microscopy, apart from looking at micromorphology and morphology, you can often also look at element composition of plant surfaces. And this is a lot more exciting than you probably think. A combination of material contrast with backscattered electrons in the SEM and EDX permits not only the identification of roughly differently structured plant surfaces, but it even permits the identification of the primary elements involved. And then with some additional analyses, you can actually identify the chemical compounds involved. In this particular case, we did an extensive study of trichome mineralization in borogenales. And we could show that even trichome biomineralization in the order borogenales shows clear phylogenetic trends. The color coding on the right-hand side is for the individual dominant elements, silica in red, calcium in green, and phosphorus in blue. And you can see that within the phylogeny of the different families of of borogenales, you find different mineralization patterns, with some of them mineralized with silica only, some of them mineralized with calcium and phosphorus only, or calcium apatite. All these studies are possible only if you've got the plants actually at your disposal in a living state, because the minerals are quickly translocated if you try to fix them in ordinary fixatives, and they are definitely no longer present in their original location if you try to use herbarium material for these studies. Maybe the most surprising result was the fact that Arabidopsis thaliana, the world's most boring plant, shows some striking patterns of biomineralization. This is a typical trichome of the leaf surface of Arabidopsis. And if you look at it more closely with EDX, what you find is that the entire trichome, apart from its very base, you can see that in the major picture, is mineralized. But if you look at the inserts, you can see that there is calcium nearly all over the trichome, whereas phosphorus, the left picture on the insert, is localized in these little granules on the trichome surface. So calcium phosphate, a plant mineral that we first discovered in Loasaceae a few years ago, has actually been hiding in plain sight on the world's most investigated plant, which means that calcium phosphate calcium apatite appears to be a common plant mineral, quite contrary to accepted wisdom that calcium phosphate plays a role only in animal biomineralization. Of course, grasses are also heavily mineralized, and one of the striking things about botanical gardens is that they tend to have very poor collections of grasses. There are a few exceptions, but grasses are generally considered as boring and superfluous, and while gardens invest a hell of a lot of time in keeping their rose borders in order, few of them have any noteworthy collection of wild collected grasses across the tremendous spectrum of diversity they show. Our studies of biomineralization of grasses cultivated at Bonn University Botanical Gardens could, for instance, demonstrate these interesting patterns of biomineralization in a coated area, a grass that you all know, one of the few that most gardens have. This one here is, however, coated area Orocana, a wild species from South America. And they are mineralized with two different structures. On the one hand side are the cystoliths, which you see on the upper right on a normal BSE image, and then on the color image, or the false color image, you can see how these cystoliths are immersed in the upper leaf surface. But you can also see that these coated areas, they've got these sharp little trichomes. And if you ever wondered why it's so easy to cut your hands on the leaves of that plant, then the reason is that they don't only have trichomes, but the very tip of the trichome is essentially a tiny little glass peak, so that the trichomes themselves are, of course, organic, indicated by the green color here, but the very trichome tip, the one that rips your skin open, is actually made of silica. Trichome mineralization is a fascinating topic, and this is just the last image on one of my favorite plant groups, the stinging nettles. And stinging nettles are, of course, notoriously stinging. And the stinging hairs have been investigated over the past 200 years, but not with the techniques available at in our times. So we looked at biomineralization patterns across all plant families that are provided with stinging hairs here. This is another example from Loasaceae, but we also looked at those of Urticaceae. And what we found here was extremely striking. The tip of the stinging hair of this particular plant species is mineralized with silica, phosphorus, and calcium. That's the three inner images on the insert on the right hand side, and we can add them up into a false color image, and then you can see that the bulk of the stinging hair is indeed made up of calcium, actually most of it of calcium phosphate, but there's a tiny thin varnish or cap of silica on the very top of the stinging hair, possibly increasing its brittleness and making sure that it reliably penetrates the skin of any animal trying to eat it. Stinging nettles are one of my favorite plant groups, and one thing that you all know about stinging nettles, especially the greater stinging nettle, is that they tend to have unisexual flowers, and typically they are dioecious. Of course, the greater stinging nettle is often has individual monoecious clones, and then both male, female, and monoecious individuals coexist, which we call polygamy. And in the literature, it has been known that Urtica is either monoecious or polygamous, but we could also show in cultivation that there are quite a few species which are either gynodioecious or androdioecious. And if you cultivate a larger number of species of Urtica from across the globe, literally from New Zealand to Japan and from South Africa to Norway, then you find that gender is incredibly complex in stinging nettles. Gender includes, gender distributions are incredibly diversified, and I've just given you a list of a few of the typical expressions that are found in the Urtica dioica group across the globe. So you have Basiandrus monesi, where the male flowers are at the bottom of the plant, but you've also got Proxyandrus monesi, where the male flowers are always at the basis of the individual inflorescence branches. That's the picture on the middle left, but you also get sandwich Monaceae, you get variegated Monaceae where the male and the female flowers are wildly mixed on the inflorescence branches, and so on and so forth. So a study of the living plant was able to expand our understanding of gender distributions in Urticaceae dramatically beyond what has been widely accepted in the literature. Of course, phytochemical studies is something that I haven't mentioned before, but we've done that extensively, and everybody has been doing that ever since our founding director at Bonn University Botanical Gardens, Niels von Esenbeck, who was a pharmacist amongst other things. Botanical Gardens actually originated from medicinal plant collections. Nowadays, with the modern analytical tools, of course, we are able to find substances that our ancestors could only dream of, and we can also find many, many different substances side by side in an individual plant species, and we can compare them across many different species if we have, ideally, living plants in our collections. And this chemical aspect brings me to my last topic, and that is GC-IMS as a typical example of a modern technology that really pushes the limits of what we used to be able to analyze and what we are able to analyze now. This is something that we recently introduced at Bonn University Botanical Gardens for a study on biodiversity monitoring, but we are now using it also for floral scent analyses. GC-IMS is gas chromatography, ion mobility spectrometry. The stacked GC columns are tiny, as you can see. I'm comparing them to Eurocent on this picture, which is familiar to most of us, to our colleagues from the UK. I'm also giving the measurements in imperial measures so that you know the size we're talking about. So the miniaturized GC-IMS is portable and battery driven. Sampling times are in the range of minutes, and the two-dimensional segregation identification of PVOC, of plant volatile organic compounds, is possible very nearly in real time. So we can actually go into the garden and have the plant where it stands and take its floral scent at any time interval we want to do. And we've got some preliminary data on daffodils. You can see you don't have to enclose the flower on the upper left. You don't have to concentrate the sample. You just take the air, you sample it for a few minutes, and then you can analyze it right in this machine. And these are just some of the preliminary results we have on daffodils. PVOC analysis in the genus Narcissus. Surprisingly, we found a relatively strong phylogenetic signal, which you can see in the color coding here. On the right-hand side, that's a cluster analysis of the different scent profiles we got, and the color coding refers to the taxonomic group the species belong to. So you can see there is quite a good correspondence between taxonomic group and scent profile. That's still work in progress. We hope to optimize that. With this very simple technique, with a very short sampling period and very short analytical times, we are able to provide a broad overview over scent profiles of plants that we have in our gardens with a minimum of technical effort, with the only caveat that we definitely need, of course, measurements of plant of the respective reference compounds. Yeah, with that, I'm at the end of my talk, and I would like to conclude that nature makes sure we don't run out of research questions. Technical progress makes sure we don't run out of novel ways of investigating them. And botanical gardens make sure we don't run out of plants to answer them with. I thank you very much for your attention, and have a fantastic 400th anniversary meeting.